Hi, good day, and welcome to our class in CPT 3 Fundamentals of Crop Science. So, for this afternoon, I'm going to explain to you in continuation of our topics on biotechnology, the genetic engineering. So, for now, since most of you and all of you cannot attend the class for various reasons, technical connections, etc., so I'll just be explaining to you in video the topics that we're going to have. So, last time we have definition about biotechnology. So, from the life study of life bio and technology, which is about all the recent technology processes. So, another definition of biotechnology is the integrated use of biochemistry, microbiology, and biochemical, genetic, and process engineering to manufacture products. So, um, by the way, this report was done by my classmate before in PhD, Miss Roselle Cruz. So this is about genetic engineering specifically. So genetic engineering is just part of biotechnology. As you can see, it's an integrated use of all the sciences in chemistry, biochem, biology, and genetics. And this is to manufacture genetically engineered products. So, another definition is the application of technology to improve the biological function of an organism by adding genes from other organisms. So, last time I've given you examples on uh, bio, biotech crops such as the BT corn, BT cotton, and so on. So let's talk about the scope of biotechnology. As you can see, genetic engineering, which is the main topic of this discussion, is just part or scope of biotechnology because biotechnology is very wide. In fact, it has certain stages. No, and as I have said before, tissue culture is just under biotechnology. The fermentation is under biotechnology. So these are the scope of biotechnology, starting with genetic engineering, which is more of very high technology, advanced uh, biotech, the biochemistry, the biology, and the microbiology. These are the scope, all scope of biotechnology. Now let's proceed with the stages. So for the stages of biotechnology development, we these are categorized into three: ancient, classical, and modern. For ancient is the early history related to food and shelter includes domestication. So as I've told you, agriculture and biotechnology are perhaps as old as civilization. When men started to look for food, domesticate them, and shelter. So that's under ancient biotechnology. And another one is classical. It's built on ancient biotechnology as well. So dito na it's where fermentation falls, which promoted food production and medicine. Now, example of that is the very old cheese fermentation, beer fermentation, and wine. So, and the last one is the modern biotechnology, which manipulates genetic information in organism. We call genetic engineering. So, how do they do that? So, we're going to talk about that on the next slides. This another stage of biotechnology development is traditional or old biotechnology. So these are the conventional techniques that had been used to produce beer, wine, cheese, and many other food, as I have said a while ago. And the new or modern biotechnology are the old methods of genetic modification by recombinant DNA or deoxyribonucleic acid and cell fusion techniques, together with the modern development of traditional biotechnology process. So if you remember in your genetics or plant breeding subjects, so you will remember about cell fusion techniques in order for breeding. So these two stages are actually like us between classical and modern, but they can also be called as traditional, old, or new, or modern. It's just the same thing. So now we go to the selected opportunities for applying biotechnology. What is the use? Why do we need to use genetic engineering? First is for crop productivity. It's in over for overcoming biotic stresses in insects and weeds for insects and weeds, and overcoming abiotic stresses in terms of soil fertility, heat stress, drought stress, inefficiency of resource use. That's why we have crops that are genetically engineered to be drought resistant, for example, or resistant against viruses and pests and diseases, like the BT corn, the BT eggplant. Now, also for the nutritional value of crops, enhancing vitamin concentrations, mineral concentration, and even protein content. For example, is the golden rice, which is developed now by IRI, or the International Rice Research Institute at UPLB Los Baños, Laguna. So, and also for plant-made pharmaceuticals for the use of vaccines. Hopefully, we gonna have the thing that we are doing now in the coronavirus vaccines is also done through biotechnology or genetic engineering. And the protein in therapeutics. Improve food security, and that is to reduce post-harvest loss, risk to food production, and alternative uses. And also biodiversity. Engineering safeguards protects existing biodiversity and preserving biodiversity, as well as natural resource conservation for the use of renewable energy and phytoremediation. So these are all the opportunities of applying the application of biotechnology. Now, if we dig into genetic engineering, we always owe this to the person who discovered genes, no? Because we'll be talking about DNA, genetic makeup here. So Gregor Mendel is the father of genetics, a monk who grew peace, and that every progeny has two copies of hereditary factors, and discovered that inheritance is based on discrete units which later called genes. So, I guess it's self-explanatory when we say that um, each progeny has two copies of hereditary factors. So, and that makes up our physical makeup, and what if for crabs, so its characteristics, and for animals and physical, both, and physiological characteristic. Good morning. So that what is DNA? DNA is the genetic material known as the deoxyribonucleic acid. It's a thin thread-like molecule tightly packed in chromosome. So this is the figure of the chromosome. So that is not seen by the naked eye, but that is actually seen under a very high technology microscope known as electron microscope, or even visuary abstract, and for genetic engineers, they are more on abstract. So as you can see here, there's are the types of the thymine, adenosine, cytosine, guanine, and deoxyribose sugar, phosphates, and hydrogen bonds. So these are the basic building blocks of um, of our genetic material. So we call them bases, which are four kinds. As I have mentioned, the adenosine, the thymine, the guanine, and cytosine. So these are the DNA, what makes up the DNA and its picture. So genes are made up of portions of the long linear molecule of the DNA that gets translated into proteins. So basically, they are all proteins. Carry the coded information about traits that is transferred from generation to generation. So that's how mysterious life is, biological thing is, because these DNA strands are even this contains all the physical characteristics and everything that are even transferred from generation to generation to its offspring. So it produces the proteins which catalyzes cellular processes that leads to expression of observable traits, or we call them phenotype, or the physical characteristic, and responsible for the varying traits among species and individuals, as I have said a while ago. So we have this called central dogma. It's the transfer of information from DNA to protein. So um, protein, we call them protein transcription and translation. So protein, as shown in the figure, is translated into RNA with mRNA, um, um, RN, RNA, tRNA are transcripted to DNA, and DNA replicates and even reverse transcripted to RNA. So it is shown on this figure here how is the central dogma. It's basically the protein translation and the DNA transcription and RNA transcription. So it's shown here where DNA put DNA or RNA are reverse transcripted to protein in this particular figure. And we have this replication of DNA where it duplicates and transcripted to RNA and is synthesized in the cytoplasm and are translated to protein for synthesis. So, making of proteins. So how is proteins made? By our physiological process. So we have the figures here as these are the bases. So in your genetics, you have learned on how these proteins are made through the different bases. So it's actually positioned as here. So you is the it's the cytosine, adenine, the guanine. So when they are just positioned like that, and then they are combined and form into these different strands. So advances in molecular biology. It is the combination of restriction or modification enzymes and hybridization techniques enable the application of a wide variety of procedures. And this includes some applications, the gene isolation or purification, the polymerase chain reaction, sequencing or genomics, the RFLP, mutagenesis, reverse genetics, cell tissue culture, and genetic modification. So, in your laboratory exercise, your first laboratory exercise is to watch videos online, or perhaps if you can, it's better to visit a PCR lab, and you're going to research and study the different equipment and the functions and all the process activities that are done there. So I hope you're starting to do your laboratory exercise 1, which is about the PCR or the biotech lab. So, evolution of breeding. So, actually, breeding is as old as civilization when we first domesticate plants and animals. So, it's the ancient art of plant breeding actually started on that time. So, this involves the principles of genetics and cytogenetics. Science-based plant breeding is through chromosome-mediated gene transfer, wherein um, people or humanity intervene with the natural plant breeding or ancient way. So, this involves molecular cytogenetics and modern biotechnology. And the recent one is the molecular plant breeding. It is the genetic transformation through direct gene transfer. So it is actually the gene that is inserted manually through equipment. So specifically genetic engineering, we define them as the methodology that employs recombinant DNA, or we call them rDNA techniques. And it's also synonymous to rDNA, gene splicing, biological engineering, or modern biotechnology. So that's all the same thing. It involves isolation, modification of genes, insertion to new species, and development of transgenes. So we call them transgenes if the genetically engineered genes is added to a species. So as what I have said, the very basic example is the BT corn, BT eggplant, or the Bacillus thuringiensis. So this allows the incorporation of the foreign DNA into the host organism where the DNA does not naturally occur. So if you want a certain characteristic like pest resistance, so into a high yielding variety, so you are going to get that DNA and transfer it to the genetic makeup of of your host plant. So, and of course, is capable of continued propagation. So, it can alter the hereditary material DNA of a cell so that cells can produce more or different chemicals or perform completely new functions. And for the development of organisms that express a novel trait that not normally found in the species. So, with this genetic engineering, it has been possible to really combine all the genetic makeups or phenological types characteristics that you want in a particular plant. And these are very common in tissue culture of orchids. So that's why you you can find a lot of varieties of orchids. So we call it the recombinant DNA technology. This involves um, isolating the gene and identifying it, coding for a particular protein, and placing it in the host cell where it can be produced. So there's need to be a host cell. So in the case of Bacillus thuringiensis, it uses a particular bacteria as its host. So it necessitates isolating and separating the messenger RNA or messenger mRNA responsible for the genetic code of the protein required for cloning. So the particular RNA is to be isolated, thus creation of a new combination of DNA molecules that are not together naturally. So these are the basic steps, although you may find this as you in your laboratory exercise, so you can maybe watch videos on how this is done in the in other advanced laboratories. It may be international or just around the country. So but basically these are the steps how it is being done. So first you have the DNA purification from cells or tissues. So you are going to have, in order to have a bit example of this practice in your laboratory exercise number 2, you're going to have the DNA extraction. So, and the steps are just there. So if you have any questions and also photos to provide you how to do it, and you can also search on videos on internet or in YouTube on how to do it step by step. So you really have to demonstrate and make a report in your laboratory exercise too on how to extract DNA and purify them. So however, the steps of um, rDNA technology so is really so advanced. So you just do the first step, and after that you will realize that when you get the pure DNA, when you have separated them, then it's not it has actually a very wide use, you know. Next step is the generation of DNA fragments using restriction endonucleases or enzyme. So there are also certain this use enzymes, but there are also ways that you don't use enzyme, perhaps machines or equipment. So restriction enzymes like the ECO R1 and so on, these are these recognize and cut DNA molecules a specific nucleotide sequence. So you will learn in your laboratory exercise that enzymes are capable, no, like capable of like for amylase, it's an enzyme that degrades amylose, no, or or starch. So next step is joining of fragment to other DNA molecules that serve as vectors or the recombinant DNA molecule. So this time you're going to join the fragment together. So plasmids are used as vectors or vehicles used for DNA fragments. Next is transfer the rDNA molecule to a host cell where it replicates and produces identical copies. So mostly the ones that are used are single-celled organisms like bacteria. And replication of host cells are rDNA passed on their progeny cells, creating a population of host cells, each carrying copies and cloned DNA sequence. So you imagine a certain bacteria then divides and replicates with with the inserted genetic makeup already. And then your next step is recovery of cloned DNA from host cells, purification, and analysis. So you also have to test if the clones, the clones copies are actually a clone DNA or it has the target physical characteristic or trait. And then transcription of the clone DNA, translation of mRNA, and isolation of the genetic gene product. So as I have said, these are not seen on the naked eye. This is being done without even seeing it, no. So it's just a process that are usually visual, no, abstract. So it has this photo figure describes how is the steps being done, you know, with the vector and donor DNA digested with restriction enzymes and then mixing the DNA ligase added and seals overhangs, and then DNA is introduced into bacterial cells. We have the recombinant DNA, molecules replicate and cells divide. So, and then you got the clones in the end. And then after that, you have the cells with the genes of interest into the bacteria, and then the copies are replicated and are harvested. or the the goal may be to make copies of genes, and then genes are harvested for for pest resistance, for example, or gene alteration. So the applications of genetic engineering is, when do we do that? If the trait is not present in the germplasm of the crop, it has been explained earlier. And if the trait is very difficult to incorporate using conventional breeding methods, like for example, sometimes for corn, for instance, if you know the that corns are open pollinated. They are not self pollinated because the tassel and the ovary don't drive. Like the tassels will 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 before the tassel will fall into the ovary of the cord, then it's too late, no, kasi the tassel will um will pollinate earlier than the ovary is receptive. It's not receptive anymore. It's so late. So there's no synchronization. So usually corn are open pollinated, or strictly open pollination. So for that instance, for a plant to be difficult to produce, so it needs genetic engineering. And it takes very long time to introduce or improve such trait in the crop by conventional methods. Like the natural breeding the natural way of breeding is done like 10 to 20 years. But for genetic engineering, it should be earlier, from less than 10 years to 5 years. So we call them genetically modified organism. These are the transgenics. So when you say the word transgenics, these are genetically modified organism or products of GE, or genetic engineering. So it refers to a living thing that has been altered by the insertion of genes to achieve tar- targeted modifications in composition or performance. So in 1997, the first genetically engineered crop was grown in large scale, where first rumblings of controversy about GMO came, and the name given, such as Frankenfood or Monsantan. So as early as 1997, genetically engineered organisms have been produced and also face a lot of controversies or debates. So there are both negative, positive perception of GMOs. So I hope to hear your reaction later on your comments on this video and questions, perhaps if you are pro GMO or not. So for the negative side, it may be hazardous, damaging, and risky, but the benefits are advantageous, and they are also saying that it's safe. Like for BT eggplant, no, they say because usually we really eat eggplant, no, as vegetable. So they say it contain the BT, Bacillus thuringiensis, that kills the insect that is damaging the crab or eggplant. How so there's also, people are thinking that there's also risk of being poisoned, from eating on that GMO crop. But then, because scientists explain that the Bacillus thuringiensis cannot thrive in acidic stomach like what human beings have, so they are not actually that cannot damage humans, no, but only for insects that feeds on the eggplant. So I leave that to your perception. So I hope you're gonna comment or share your idea on that. So these are likely to purchase biotic foods for specific benefits. Like for the majority of Americans would be likely to purchase foods from plants produced through biotechnology for specific benefits. So this study has been done before, and it shows that like in 2000, from 2006 to 2008, which provide more helpful fats. So um this gray very likely, and this side here are somewhat likely. So you can see that somewhat likely has more percentage compared to very likely. So this will show that because this is done with the majority of Americans, whether their choice on reducing saturated fat content, pesticide reduction, taste better or fresher, avoid trans fat. So for them, there's also a sign of acceptance. But for now, that was way before, no. But for now, um, we are even using the BT eggplant now and BT corn, no, in even in the Mindanao region, because it was first distributed in Luzon area, but now actually, if you're going to ask a lot of farmers now are actually using the the round ready, the RR corn, like something like that. So what are the applications of genetic engineering in foods? So there's a lot, like for cheese production, and so for cheese production, so I will not go into details. So how this does um because it's everything explained here. It's even the enzyme that has being used. So they use genetic engineering in milk production using casein that that is used for cheese production. So chemosin has been also used as a digestive enzyme for the for coagulation in milk through the hydrolysis of casein. And it breaks down the milk, causing them to clump together and separate from the watery whey. So, um, before during 1960s, there's had been shortage of brunettes. So they use this and also the age of cow will affect chymosin production in males. So they use recombinant DNA technology to use this certain bacteria and fungi, the Saccharomyces these are fungi, Saccharomyces and Lactis, Aspergillus niger, Trichoderma, as host for the chymosin. So this has been modified organism which contain the gene derived from the stomach of cows responsible for the production of chymosin. And this has been approved by the US FDA and became the first product of genetic engineering. So the new genetically engineered cheese rennet are actually 100% identical to chemotrypsin, high yield, more firm, more mature, and preferred flavor, and is toxin free. So we don't know actually now we are eating this kind of cheese, because especially when they are imported from other countries like US. So differences attributed to the presence of trace amounts of proteolytic enzyme in the recombinant enzyme which remain active during cheese ripening. So another use is for starch processing. So There are sugars like glucose and fructose used in food processing, and these are produced from starch sources such as corn, wheat, and potato. And it has enzymatic processing enzymes like the amylase, the glucoamylase, the glucose isomerase. So the gene encoding for the thermostable enzyme are being used, the alpha amylase from Bacillus stearothermophilus and the glucose isomerase from Thermus thermophilus. So using that, enzyme complex is active at high temperature and increase in the rate of fructose and glucose formation by approximately 16 times and 6 times respectively. Okay, another is for brewing or beverage processing. So for in beer, this is produced by fermentation extract from malt or barley with aromatic herbs and hops for flavoring. So with the use of GE techniques, they modify yeast strains to enable them to ferment rapidly, tolerate high ethanol concentration, increased number of metabolic sugars, and metabolic byproducts. So they use recombinant DNA methods such as the ability to degrade various carbohydrates and proteins, modify the process responsible for beer flavor production. So, with two criteria of good strain of yeast, stable under fermentation condition, and produce sufficient. So, they use the glucoamylases, the brewing yeast, dextrine, glucoamylases from Aspergillus niger, and production of yeast strain capable of producing its own glucoamylase. So, with this rDNA technology, through transformation of yeast strains with gene for a glucoamylase, the enzyme capable of degrading the normally unfermentable wort dextrine to glucose. So, this is called the DEX1 or STA2. The genes for this enzyme are cloned from the uh, Staphylococcus cerevisiae and inserted into the plasmid containing the bacterial and yeast DNA. So, also lacks this debranching activity with considerable amounts of wart dextrose. So they also use the GA gene from Aspergillus niger, which encodes this glucoamylase with both this hydrolytic activities were cloned and inserted into a vector. And the H2S hydrosulfide production, yeast produced during some fermentation is removed, and transformation of brewing yeast with this STR4 gene, which produces less H2S than transformed strains. So using STR4 gene, it enhances the cystathionine synthesis and thus removes excess H2S. So for the production of flavor compound, this also has been used, no? The sulfur dioxide in beer, which is responsible for flavor stability, antioxidant, and sequestering carbonyl compounds or the stale flavor. So we have the MET3 and MET4 genes for encoding ATP sulfurylase and APS kinase. These increase the flux through sulfate metabolism pathway, thus producing high levels of SO2. I know this is been very technical to you, but this is just to explain you how the genetic engineering of, for instance, brewing or beverage processing, such as beer, has been done, and specifically and they also do for copper resistance, problems on sensitivity of brewing yeast to copper ions. So several things of uses used in brewing industry are transferred using plasmid carrying to the dominant gene copper resistance, known as the COP1. Gene codes for copper chelating protein, which produce high number of copper chelating protein. So there's actually a lot of other transgenic products of industrial application. For example, human milk proteins such as lactoferrin and casein in transgenic potatoes can be used as supplement in human infant formulas and baby foods. Another one is the monoline in sweet protein, which is 100 times sweeter than sucrose from plants expressed in lettuce and tomato. So this can be used to replace sucrose, which is extensively used in confectionaries and other products. So in agriculture, crop production naman, tools used for plant improvement, which has, as you can see here, this is the history on how biotechnology has been used, and uh, the it's even 100 or 12,000 before Christ. No, wherein grafting is first used, or plant selection. But with the use of hybrids from 1694 to 1900, so with the sexual reproduction, the first intergenetic cross, the hybrid maize production, which began in U.S., and mutation occurred from 1909 to 1967. First, protoplast fusion, X-ray mutation breeding, plant regeneration from isolated cells, and recently from starting 1973 to 1994, where where is the first GM food approval. These are all under genetic modification through biotech, wherein the first recombinant DNA molecule and genetically modified plant. So this is just to show you the breeding methods in comparing the traditional plant breeding and the modern plant breeding. It's actually all it's just the same. Now this is the conventional, all the traditional, but with the genetic engineering, there is the insertion of particular genes steps. So in simple terms, uh, we call the the product through of genetic engineering as transgene, and we call the process as transgenesis, wherein the genes of interest are inserted into the host plant. And now with a combination of two different varieties, we have this is for traditional breeding. For crop production, it has a various uses, no, to improve yield, productivity, quality of plant crops, and nutritive quality. So once again, this includes tissue culture, somaclonal variation, somatic cell hybridization, and rDNA. Don't just um, get wrong with these terms, because these are actually the technologies that has been used in genetic engineering, which are very advanced. The first commercialized GM is the tomato flavor savoir, which show delayed ripening. Other GM food crops such as soybean, alfalfa, or the alfalfa is the oil, canola oil, and sweet corn, all possess the resistance to insects and tolerance to herbicides, like the Roundup Ready corn. Identification for the steps, um, it's it's just the same with the steps that has been explained earlier. So first, identification of the genes responsible for producing the desired characteristic into the donor organism, which is usually bacterium, and isolation of the gene interest from the donor organism, and addition to the appropriate gene switches to the gene of interest. So these are categorized into steps. First is the insertion of the gene into the cells of the host plant, of the host organism, which is plant, and enucleation of the transformed cells into the selected media. So this is being reared also in a tissue culture way, and the application of conventional breeding for seed production. So this is now, it also involves the breeding or seed production. So this figure will explain the different processes. First, we have the plant chromosome that with a transgene, and then and incorporate them through tissue culture techniques, co-cultivation and selection, and plant development. We have the transgenic plant. So what are these herbicide-resistant crops? For you all know that glyphosate is a broad-resistant herbicide, which is an active ingredient in Roundup herbicide. So it kills plants that made contact with the inhibition of the key enzyme EPSP synthesis in the amino acid pathway. So definitely plants die due to the lack of this amino acid because it will block the enzyme. And a resistant EPSP synthase gene will allow the crop to survive for spraying. That's why the Roundup Ready corn, as shown on the figure here, the shikimic acid pathway plus phosphoenol pyruvate with glyphosate, it blocks the EPSP synthase, thus the and without the amino acid the plant will die using the Roundup, no, because it has the glyphosate that will kill all plants, you know. But with the Roundup Ready resistant plants, it has with the shikimic acid and phosphoenol pyruvate, it has the bacteria APSP synthase, so Roundup has no effect enzyme in resistant to herbicide. Thus, we have the aromatic amino acid as products, so the plant will live. So if you in the field, if you're going to compare the herbicide resistant crops before and after, so it will not die. So if you have you comment on in this video if you have tried to use an herbicide before or a Roundup herbicide. So if you're going to spray herbicide on a Roundup Ready soybean or corn, it will not die if it is RR corn. No, but if it's not an RR corn, a Roundup Ready corn, it will definitely die using herbicides. So in crop production, genetic engineering is also used in minimizing crop losses due to the pest, weeds, pathogens. So crop losses rate and potential loss is higher, in these crops. So this is the reason why we use genetically engineered crops. But the top two reason for biotech crop adoption is to increase from 59 to 79% increase yields through improved pest control, and 15 to 26% to save management time and make other practices easier, and 9 to 17% decrease pesticide costs. So if you're gonna use RR corn, you're or you use BT corn, you're not going to spray pesticides, but also for Roundup Ready corn, is the labor for weeding is cut off. You just spray them with herbicides. So another for post-harvest naman is delayed ripening. For example, for tomatoes, although ripening is a natural aging or senescence process that involves two independent pathways for flavor development and fruit softening. So typically, tomatoes are picked when they are not very ripe to allow safe shipping of the fruit when they're still a pinch of yellow or red, but not totally green. Polygalactorinase, the enzyme that degrades pectins and plant cellulose, contributes to the fruit softening. So what they did actually is they do a polygalactorinase gene expression. It was inhibited by the introduction of antisense polygalactorinase gene. So they call it antisense polygalactorinase and created the first commercial genetically engineered plant called the flower savoir tomato. So it has been mentioned earlier that the flower savoir is the first genetically modified available modified tomato or modified product that is has been available commercially. So it has, please remember that it has the antisense polygalactorinase gene in order to delay its ripening, thus prolong post-harvest shelf life. So another one for ethylene production for pear, which is normally harvested at preclimacteric stage. So the ethylene production at this stage bursts and results in flesh softening and synthesis of aromatic compounds. So to reduce shelf life and quality deterioration, they use the ethylene biosynthesis pathway from methionine, or some we call that ACC, into ethylene. This is a normal. So there are two enzymes that catalyze the pathway, the ACC synthase and the ACC oxidase. So these cDNAs corresponding to mRNA of both enzymes were cloned for many plants. And these transgenic plants with reduced ethylene production were produced using antisense genes, the same with the tomato flower, or sense co-expression for ACS and ACO genes. And so the production of ethylene supports transgenic plants. And also they use this for nutritional enhancement. For potato, which is deficient in lysine, tyrosine, methionine, and cysteine, they improve the nutritional value through transformation with non-allergenic seed albumin gene, AMA1. So this AMA1 from Amaranthus hypochondriacus, producing a well-balanced amino acid composition with no allergic properties. And also inulin to reduce the energy density of food and used to enrich dietary fiber or replace sugar and fat in foods from artichoke. And for tomato, it has developed high caro or HT tomato. HT is a transgenic tomato expressing the tomato lycopene beta cyclase, and it produces an orange fruit due to complete conversion of lycopene to beta carotene and has averaged 76 UG beta carotene, 7 times more than the non-transgenic. So it can supply 3 times the adult RDA or recommended dietary allowance for beta carotene. Another one is biofortification. So it is the biofortification is the process of producing crops that are rich in bioavailable nutrients and can be through conventional plant breeding or biotechnology. So, but compared to conventional breeding, transgenic technology as part of the tools of biotechnology were touted to be more efficient method of improving specific crop traits and as well as introducing novel characters. So biofor, biofortification of staple food is food grass is a new public health approach to control nutrient deficiencies in poor countries, and it predominate the diets of the poor. One-time investment to develop seeds cost effective, and biofortified crop season is highly sustainable, grown year after year. Feasible means to reach undernourished population in relatively remote areas. So, with biofortification through genetic engineering has been the hope for poor countries or under hunger. So in Philippines, this has been developed, the biofortification of rice. The worldwide consumption of rice is 423 million tons as reported in 2008. And in Asia, 377 million tons are consumed, which is 89%. With beta-carotene is not naturally present in rice, is the major staple food in Asia, and hence the use of biotechnology is actually required. So how does this created? So Ingo Putricus, a German scientist, in 1999, together with the staff, create a rice variety that produces molecules that imparted yellow color in seeds known as the golden rice or GR. So Putricus and Peter Bayer worked for the pathway in the possible genes, and the genes involved were from daffodil and bacteria, and complete the pathway for the VA synthesis. So they explain how to do it in these figures. The beta-carotene pathway problem in plants compared to the golden rice solution. So as you can see here, that this is the normal steps in the beta-carotene pathway, and they incorporated it with the rice. So to just illustrate you the different steps, so from IPP, then geranyl general diphosphate, phytoin to lycopene to beta-carotene, but then rice lacks these enzymes. So normally, rice is vitamin A deficient. But with the golden rice solution under the beta-carotene pathway, genes added from IPP, geranyl diphosphate, with the daffodil gene, and it is incorporated already into phytoin, into lycopene, into the beta-carotene, and with the daffodil gene, the golden rice is created. So this pathway is being modified with a single bacterial gene perform both functions. For the vitamin A pathway is complete and functional. So if you have questions, or you can always comment on this video for your questions regarding this illustration. So for the golden rice development until now, ever since, as you know, as mentioned, it's still from 1999 until now, it has not been commercially produced in the Philippines, no, or somewhere, but it's still under research. So current carotenoid level of this golden rice ranges from 1.6, and need to consume 3 kilos of rice for this 1 to 3 years old child to meet the RDA. So for the carotenoid level of this GR gene, PSY gene from corn, with 14 UG per grams, 80 to 90% is beta carotene and weight of same as non-transgenic. And for carotenoid level of corn, PSY plus the bacteria, it has 37 micrograms per gram. With 31 microgram per gram beta-carotene in polished rice, and a consumption of 72 grams of dry rice day would suffice for a child. So the field trials are conducted at Erie last April 2008, and with the content of beta-carotene, high yield, seed quality, and appearance. So this is the photo that will show the wild type rice and with the daffodil and corn. So this figure will show you the generation of biotech crops. The first generation from 1994, the second generation in 1995, and the third generation 1998. As you can see, most on the first generation of biotech crops are mostly herbicide tolerance, insect resistance, virus resistance, disease tolerance, and delayed ripening. But on the second generations, more on high nutritional value, amino acid rich, premium quality, and low allergenicity. And on the third generation are vaccines, antibodies, pharmaceutical proteins, abiotic stress tolerance, and plant-made industrial products. So, concerns of genetic engineering, engineered foods. For producers, the impact in the market, but now I think it's a less problem now because people are being it's been introduced already and widely informed. But the cost of production is a problem, and the production yield. For consumers side, the sensory qualities is the main concern, and the price. So cars produced through biotechnology undergo an extensive safety assessment and scientific reviews before being commercialized. That's why it takes at least 10 years or more to develop this, because it contains safety procedures such as the comparison of GE properties with the conventional products, safety of new proteins or compounds novel to the crop, comparative analysis of known toxicants and other nutrients, and safety assessment using animals or livestock feeding studies. So these are the very detailed, I would say, explanation of genetic engineering. So thank you for watching this video, and if you have questions, just comment your question, and if you have ideas to share, you are very much welcome. So thank you, and I will see you on the next meeting or on the next video.